Hi, my name is Martín Holguín Osorio. I'm an ichthyologist and a master's student from the Antioquia University. The title of my work is Detecting Fish Diversity in the Andean Rivers using Water Environmental DNA Metabarcoding, eDNA. The inventories of fish diversity are done by methods of traditional capture, which are limited, invasive, and lethal. One alternative of raising interest is the metabarcoding, which allows us to rescue and analyze the DNA sequences released from the fish to the water in the environmental DNA. A method was implemented and optimized to the Andean conditions in order to identify the fish species through metabarcoding in different Andean flows with the presence of reservoirs. Once the water sample was taken, we proceed to filter it at the camp, not without before prepare the work area and the equipment that were going to be used, given that the cross-contamination between samples should be avoided at all costs, since the high sensibility that metabarcoding technique has. The filter, which is the warehouse of the environmental samples and has all the info that we're searching for, was stored and transported to the lab where the posterior eDNA extraction was done. The amplification, NGS libraries, and NGS sequencing was done by hiring an international lab, due to there's a lack of these sequencing technologies in Colombia. Once the data arrive, we proceed to do the bioinformatic process to analyze it, which was a challenge to me. For the first pilot assay, we used dolerite primers, which are for metazoans in general. Here we did just find 2 species. Also, there were very few DNA from fish, just 0.86%, due to the very high variability on the streams of the rescued DNA segments or the anchor region of the primers. In order to take more fish DNA, we optimized the primers by reducing the variability on the anchor region of the primers. Having a more conserved anchor region allowed us to capture more DNA from fish, 40.2% versus 0.86% of the first assay, and have a better detectability rate, more identified species. With the help of in silico PCR with complete mitochondrial genomes, we could design and test by simulations and without any other pilot assay a new couple of primers that aim to the 12F gene, where strings of the rescue sequences are conserved as possible into the group of interest. In the first sampling, we detected just 2 species with the koi primers in just one environmental sample. Also, they didn't amplify in all the environmental samples. While, on the contrary, the 12S primers did amplify in all the samples and detected a species. The respective environmental phylogenes did show something similar. For the second sampling, we started to take 2 samples of 2 liters. With the koi primers were detected a species, and again were some samples that didn't amplify it at all. The 12S primers have a better performance again, and the situation with the phylogenes was very similar than the seen before. In the last sampling, the koi primers have the worst performance that we've seen, finding any species, while the 12S primers did find 12 fish species. With environmental phylogenes, it's clear that the rescue diversity for the koi primers isn't the best to identify fish in environmental samples. For last, we did a positive control, and again the 12S primers have a better performance, finding 40 fish, while the koi primers could find 22. So this reinforced the idea that the rescued diversity from the koi primers wasn't the best to identify fish in environmental samples, and even when there's just fish DNA, because this was a bull sample with only DNA from 42 fish different species. So the choosing of the primer is critical if we are going to search fish in eDNA.